All right, everyone, I'm going to have you go ahead and get seated. We're going to go ahead and start the program today. Before we get started, there's just a few things. First of all, we have, to save paper, we have our program via electronic form today. So if you want to go ahead and take a picture of the QR code and want to follow along with the program today, that would be great. Before we get started, we always have our land acknowledgement. And we have a student here today that has been graceful enough to accept my offer to do so. And her name is Kateri Phillips. So if you could come up here. Katari, excuse me.
Hello, everyone. Before I give this acknowledgement, I would like to do a traditional introduction as that's common practice in our local Anishinaabe communities. Buju, Bidore Benisike, Indigenous, Bishna Chiga, Nina Dodem, Bodewada Miandao, Minwa, Shishibani Gatamatagoza, Miyagi Anishinaabe King and Dochpia. Hello, my name is Kateri Phillips. I am Potawatomi and I belong to the citizen band Potawatomi Nation, which is a little outside of Shawnee, Oklahoma. I'm also the current president of the American Student Association, and on behalf of Northern Michigan University, I would like to acknowledge that we are on the homelands of the Anishinaabe, namely the Ojibwe, Potawatomi, and Odawa nations. Mikach, Mnogishet Nangom, thank you. Have a good day, everybody, and good luck, presenters.
Welcome, I'm Lisa Eckert. I'm the Dean of Graduate Studies and Research. My job here is limited. You don't want to hear me talking. You're here to hear the students. And so we're looking forward to that. But I would like to first thank our judges. We want a round of applause. We could not do this without them. They're taking time out of their busy days today. And when I say your name, if you'll please just give a wave to everyone so they know who you are. First, we have Adam Levine from the National Guard. He is a primary prevention specialist and chaplain. So thank you so much for being here today. Liza Fust from Cal Morris Incorporated. She is the Director of Operations. And again, thank you so much for being here. Trish Davis, Kendricks Bordeaux, Managing Partner and Attorney. We have Andrew Rickauer, who is from the United Way of, I said that right, United Way of Marquette County. So thank you very much for being here today. And Rick Vanderveen of Mackinac Power. So thank you to the judges. You are in for a treat. I'd also like to tell you this is my favorite event because there's so much energy and it moves fast, and we spotlight the students who are doing such amazing work and moving their research forward. You get a little taste of it today in their 3-minute elevator speech, but we're looking forward to seeing that. An event like this takes a lot to put together, so I have to thank a few people. First of all, I would like to thank Madeline McCurdy, who is our student worker in the College of Graduate Studies and Research. Tyler Thompson, wave. Tyler Thompson is our administrative assistant, makes everything happen. I'm saving you for last. Where's Tess? Tess Sayin, there's Tess. Tess Sayin is the director of the Office of Sponsored Programs and Research Administration here at Northern. Couldn't do this without you either. Lindsay Haukala, who is single-handedly running the McNair program, freshman fellows, and the undergraduate research fellowships. She's an integral part of our team. Also, I have Enrico. Where's Enrico? Enrico Sassi in the back, who is the assistant director of the Office of Sponsored Programs and Grant Administration. And finally, last but not least, the MC 3M tier extraordinaire, the one you've all heard from numerous times over the course of the last few weeks, and that is Sarah Amadon. And that is all I'm up here for, because as I said, you don't want to hear me. I could go on all day. I will instead turn the mic over to the guide for the day, Sarah.
Oh my goodness. Thank you for being here. I'm extraordinarily nervous. So oh, look who President Tessman joined us today as well. He snuck in the room. So everybody, let's give President Tessman a round of applause for coming. So like I was saying, I am nervous as well. So to all of our presenters today, it happens. This is my first time up here and presenting this program as well, but I'm so excited to be here. My name is Sarah Amadon. I'm the Assistant Director of Graduate Student Affairs here at Northern. I have been in this position since November, but I have been with Northern for 6 years in various positions. I know Lisa said thank you, but I have to thank my entire team. Wow. This was a lot to put together, and I could not have done it without all of the help of the people in my office. See, I'm nervous. You can hear my voice shaking. So thanks for your patience and thanks for your grace for helping me out. I want to just say to the people who are Zooming in, please let us know via the chat function if you're having trouble hearing or seeing anything. We do have somebody who's monitoring that, and that would just be nice to know. I do want to point out also that this event encompasses all of some of each of the parts of the College of Graduate Studies and Research. Our office functions as the administrative, academic, and professional development support for all of the graduate students from the time that they get here to the time that they leave. We also have our office also includes the Office of Sponsored Programs, and also includes the McNair office as well. The Office of Sponsored Programs plays a role because some of the research that you will be seeing here today, if it had human subjects or animal subjects, you need to get approval for that. So that's how they play a role as well. And then of course we have our McNair students participating today. So you might be asking yourself, what is 3-minute thesis? I have tried to send out the information to people. I think it's a super cool, I just think it's super cool. 3-minute thesis is a research communication competition that was developed by the University of Queensland. And it was actually developed during a drought. So they were having a drought in Australia and they had 3 minutes to shower. And the dean of that, the University of Queensland, the dean of the graduate college, was taking a shower one day. And he thought, how cool would it be to present our thesis in 3 minutes? Thus, 3-minute thesis was born, which I think is the coolest part of that story. So it challenges students to consolidate their ideas and their research to a non-specialist audience. So they're supposed to put it in layman's terms so anybody can understand it. It is now held in over 900 universities and institutions across more than 80 countries worldwide, and we are lucky enough to be one of them. Okay, so the competition rules, presenters are allowed one static PowerPoint slide. There's not supposed to be any movement, any transmissions or transitions, anything like that. There's no additional electronic media, so they can't have any sound or video. There's no additional props. They can't have costumes or musical instruments or laboratory equipment. Presentations are limited, like I said, to 3 minutes maximum. Anybody exceeding 3 minutes is disqualified. Presentations are to be spoken, so you can't sing it, you can't rap it, you can't say a poem. They're to commence from this stage right here. And presentations are considered to have commenced when the presenter starts his presentation through movement or speech. And the decision of the judges are final. So thanks again to our judges for being here today. And I won't go through all of this judging criteria with you, but basically students are judged on their comprehension and content. You know, did they give a good understanding of what they're presenting? Did it follow clear and logical sequence? Were they careful not to trivialize or generalize their research? And did they convey enthusiasm? So all those things will be laid out for the judges. Okay, I did forget to mention, in the event of a tie, I have selected a tiebreaker judge. And that person doesn't know who they are, but in the event of a tie, we will look at the scores from that particular judge, and whoever scored higher with them will be the winner. That's how it will be determined. That's how it's determined at nationals. So that's how we will do it here. We have a Q&A session at the end while we're deliberating and doing all of the scoring. And we also have people's choice. So after our McNair students present, which they will be going first, we will have an opportunity for people to choose who they like or who they thought presented best out of the McNair students, and then we will have one for graduate students. We also have crowd trivia so that when we have our judges who need to have time to deliberate, we can have some fun and have some trivia. So we will be having our Zoom members join in with that along with the crowd here. And if you want to go ahead and click that QR code, that would be great, because that's for the trivia, which Tyler worked so hard on. And then we had a bunch of mixing around to do today. So now I would like to introduce Lindsay Haukala, who will tell us about the McNair program and start us off with our McNair presenters. Thank you.
Thanks, Sarah, and thanks for everyone who's here just supporting research on campus. As you may have guessed, since our undergraduate researchers are presenting research, McNair is an undergraduate research program here on campus. We're small and mighty today with our two presenters, and small and mighty on campus. We normally serve 28 students. So we're really able to have that one-on-one relationship with our students and one-on-one relationship for them with their faculty mentors. That's one of our major selling points we have. We also do workshops, different professionalization opportunities, and travel to campus visits and conferences to present their work like they're doing here. So today is just a little baby step in that experience, though they have presented elsewhere already, so they will be so good. Look forward to this. And the general goal of the program is to increase PhD attainment. There are 206, very specific, across the United States and 7 now in Michigan. Last go around, Michigan Tech was awarded. We beat them for many years. We've had one since 2009. MTU, boo, they just got one last time, but we're excited to work with them too. So we're starting in reverse alphabetical order. So we will start with Caitlin Younglove. And she is presenting on impacts of urbanization on synanthropic species among an urban wildland interface. Oh yeah, wrong. Spoil. Oh, you're doing it.
I'm doing it. Sorry, there you go.
Good afternoon. My name is Caitlin Younglove, and I'm here today to share my passion for protecting the species of Marquette, Michigan. I'm going to walk you through my research in 3 minutes in 3 steps. First, my methodology. Second, my results. And third, I'm going to discuss why this matters to not only the scientific community, but to the members of Marquette County. First, my methodology. I studied how synanthropic species were impacted by urbanization in Marquette, Michigan. Synanthropic species are any species that can thrive around humans. For example, think of deer, rabbit, squirrels, etc. To do this, I deployed a total of 10 cameras, 5 in urban areas and 5 in wildland areas. Second, my results. I obtained a total of 402 images of animals. 304 of those were from the urban environments, and 98 were from the wildland environments. Now that I had that data, it was time to compare. I found that there were more synanthropic species in the urban areas, which was to be expected. In the wildland environment types, I found species such as the American marten and the gray wolf, which makes sense for Marquette County. On top of that, I calculated species diversity. Species diversity measures the different types of species in an area and the prevalence of those species. I found that species diversity was higher in the urban areas. Third, it's time to talk about why this all matters. Because I found that there was a lower species diversity in the wildland areas, That led me to believe that that environment type was also suffering from a low biodiversity. A low biodiversity will impact an ecosystem's ability to function effectively. I also noticed that non-synanthropic species behaved as expected. They avoided humans. For example, the gray wolf was found only in the wildland areas and not on urban cameras at all. This led me to believe that species such as the gray wolf are staying in their habitat types, even though they are becoming increasingly smaller every year due to urbanization. I also noticed that there were no red foxes in the wildland areas. They were strictly in the urban areas. This is cause for concern because studies have shown that foxes that reside in urban areas are more likely to experience mange fatalities, mange being a parasite caused by a parasite that causes skin issues. This showcases that urbanization is pushing animals into urban areas, which is not only harming their health, but it is also shrinking the non-synanthropic species environment type that is getting smaller every year, and they will eventually have nowhere to go. These types of monitoring surveys are important for understanding biodiversity loss and how to mitigate that. Wildlife managers can take this data from studies like these and protect the species that call Marquette, Michigan home. Thank you.
Holy cow, way to start that off. Way to go. Okay, so I am your, what did we say? Your TCO, your trivia. What was that? Yeah, there we go. Your trivia chief officer. That's my title for today. I forgot it, but you've got the question already here. So which animal uses spiracles to breathe? Ants, jellyfish, sponges, or tapeworms? And please go ahead and answer online on the trivia. I will ask the audience also, just because it's more fun to hear from you directly. Just a few more seconds. All right, we've got somebody here on the second row that's got a pretty good idea. What do we think? Yeah. Ooh, that's not right. Does anybody else know the answer? No? Okay, the answer is A. Ants breathe through spiracles, much like most insects, including many species of arachnids. So our second question, which fruit is actually mildly radioactive? Are we talking apples, blueberry, mango, or banana? Oh, you know what's interesting? I just found out my son is allergic to this. I wonder if the radioactivity has anything to do with it. All right, and the answer is, does anybody know? D. Who said D? All right, there we go. Bananas due to containing an isotope potassium 40. Although the same isotope may be found in beer as well. So I guess we'll find out if he's allergic to beer. Huh? He's only 4, so it may take a while. So our next question, monarch butterflies, the only migratory butterfly, can travel approximately how far in a single day? 50 miles, 1 mile, 10 miles, or 100 miles? Okay, while you guys are thinking, I'm going to share a little personal story. I used to live in the panhandle of Florida on Eglin Air Force Base. And it's one of the largest by landmass Air Force bases in the world. And they have a lot of just like wooded area, which you wouldn't necessarily expect to see in the panhandle of Florida, but really beautiful. And there's this one space between a couple of airfields that had these really tall trees and these beautiful vines growing over them. And it was one of the places in our country that the monarch butterflies actually pause on their migratory path. It was so gorgeous to see. Okay, we'll get to asking or finding out the answer. Does anybody know this answer? Toss it out. Yep, you got it. Let's say it louder. There we go. 100 miles. Scientists now believe the larger white spots help create lift, carrying the insects on eddies that help with drag and airflow. Alrighty. And I think we're into the next presenter. I think, here we go.
So now we have Sabrina Matta, who is presenting on the interactions of Botrytis cinerea with rad. It's super rad. She gave me this hit. Tubular and rad. Radula complanata.
Came out a little weird, but it's fine. All right. So Radula complanata is a liverwort, which is a plant like a moss, and although it is over 500 million years ago, it shared a last common ancestor with the flowering plants. It is still capable of producing some very complex, specialized compounds. What's also very interesting about Radula is that its endophytes, which are microorganisms that live inside and cooperate with a plant, are some of the same species as those that are in cannabis. And this is notable because Botrytis cinerea, which is a fungal pathogen that infects over 200 species of plants and is capable of becoming resistant to fungicides very quickly, is also capable of infecting cannabis. So with our research, we have isolated endophytes from Radula complanata through a surface sterilization method that kills all microorganisms on the exterior of the plant and leaves those only that are protected inside the plant tissue to be extracted and then grown individually. And then once we have isolated them, we sequenced a small piece of their DNA to identify specifically what species we're working with. Once we identified specifically what species of endophytes we got from Radula complanata, we are going to co-culture them with Botrytis cinerea. And then once we see whether or not they are capable of inhibiting the growth of Botrytis cinerea, we are then going to investigate specifically what compounds is the endophytes from Radula complanata producing in order to inhibit the growth of Botrytis. This is important because if we find that the endophytes isolated from Radula complanata are capable of inhibiting the growth of Botrytis cinerea, this could be a great lead as a potential biological control agent. Now, biological control agents are microorganisms that are beneficial to plants, and they can help suppress or kill pathogens and pests to plants. They do this by producing toxins, starving the microorganisms of nutrients, and also by kind of helping boost the immune system of a plant. And what we hope to see is that this is an area still undergoing a lot of research, but a biological control agent can potentially be a more viable option than using chemical fungicides because as Botrytis cinerea is very capable of becoming resistant to a chemical fungicide in a relatively quick amount of time, we can instead put a microorganism on the field of plants that are, say, cannabis plants are infected with Botrytis cinerea, which will help boost the immune system of a plant. It'll help produce toxins that'll fight off the pathogen for the plant. And instead of us continuously synthesizing chemical fungicides that are harmful for the environment, we can instead use a naturally occurring microorganism that is already in the ecosystem, put them on the plant, they'll colonize, and it's a much more cost-effective measure and will be much more sustainable. Thank you.
All right, here you go. You can see the next question. What shipwreck was found in the 80s due to a secret Navy mission to find two sunken Cold War submarines? Was it the SS Thistlegorm, the RMS Titanic, SS Yongala, or the MS Mediterranean Sky? All right. Do we have any Navy buffs in the audience by chance? No? What did I hear? Someone taught that would be not correct. A, Thistlegorm, the SS Thistlegorm. Let's see, let me give you a little, oh, no, I'm sorry, I'm wrong. You were right. I was looking at something else. This guy's back here like, hold on. Yes, they both start with a T, right? And they've got an I in there. So after locating the US thrasher, the US scorpion, oceanographer Robert Ballard had 12 days remaining in his Navy mission, in which he followed a debris trail leading him to the RMS Titanic. Interesting. Okay, so our next question, who was the first woman to earn the Nobel Prize? Was it Marie Curie? Was it Lillian Gilbreth? Edith Clark? Rachel Carson? Okay, and this time I will accurately recognize the person that gives me this answer. I know there's got to be somebody out here that knows this one. Anybody? What do we hear? You got it. Hey, Marie Curie. She was the first person to win 2 Nobel Prizes and the first to win the prize in 2 different fields. All right, so our last question this break here. What is it called when an individual does not offer to help someone in an emergency if there are other people present? Is it rude? Is it bystander effect? Is it passerby effect? Or is it negligence effect? Do we have any psychological scientists that know this? Oh, because you don't have to be a psychological scientist. All right. That would be correct. Yes. Popularized by social psychologist Bib Litini and John Darley, 2 factors are attributed to this effect: diffusion of responsibility and social influence. All right, here we go. Our next group of presenters.
Yes. So we are done with our McNair portion. So you can take the QR code and it'll let you vote between the 2 for who you think is the best People's Choice selection. And we will also take a short 5-minute break for you to replenish your brownies and for the judges to judge. So we'll be back shortly.
What are you doing? Walking around?
presenters in just a few minutes. So if you guys could take your seats and make your way back, I would appreciate it. Oh. Okay. Okay, is this working? Is this working? There we go. I am so glad I didn't talk more loudly into this. So we're going to go ahead and get started with our graduate presenters. I do want to say, I know you may have noticed the last two presenters, the slide went dark afterwards. I asked AV about that, and there's not really much we can do about it. It's something that happens at the Northern Center. So today we are going to go with the flow. And if that happens to you, I will try to get it back up and running as soon as possible. But I apologize because I understand that that might throw you off a little bit. So judges, just please keep that in mind when it's going dark. It's something to do with the technology in the building. So don't hold it against our awesome presenters, okay? So first, to get started, we have Marissa Berkman. And the title of her presentation is When Floxing Flukes, Categorization of Neurophysiological Abnormalities in BDNF Floxed Mice. Here you go.
I think Bruce is gonna help me, but he doesn't know that yet. All right. Can I? Good to go? All right. Everyone knows someone here with obesity, cancer, or heart disease. If not, then you know someone who's aging, and you yourself are experiencing this phenomenon. What do these all have in common? All of these are research areas that use transgenically modified animals. If a researcher wants to study a gene, they can remove it from an animal and then study that animal's phenotype. One way to remove a gene is with Cre-Lox recombination. If the genome were a bookshelf and the books were a gene sequence, then LOXP, shown in light blue on the screen, would be the bookends that you set on either side of the books you'd like to donate or the gene to be deleted. When the Cre recombinase enzyme recognizes these LOXP sites, it snips out the intervening sequence, leaving a knockout animal. This is a common phenomenon that's been practiced in labs around the world for decades. But what if it has flaws? Brain-derived neurotrophic factor, or BDNF, is a protein that helps synapses grow and function. We study it because it's involved in ALS, which you guys might remember from the Ice Bucket Challenge. However, when we added LOXP inserts around the BDNF gene, our mice showed muscular neuropathy, increased aggression, and extreme hyperactivity. They're more anxious, obese, and they have really high serum levels of metabolic regulatory hormones, Oh my goodness. This questions the validity of these mice. It makes us ask, is LOXP really a benign insert, as we'd always thought? And if not, does that call into question all of the research that we've pulled from these labs on BDNF? One way to validate these mice is through a pre-pulse inhibition test. Pre-pulse inhibition occurs when a loud startle pulse is immediately prefaced by a soft pre-pulse. Normal brains are so good at gating out unnecessary information that they don't really react too much to the startle pulse because they've learned from the pre-pulse that, oh, that's just noise. Our mice overreact. That's an endophenotype that is consistent in human diseases like OCD, Tourette's, and schizophrenia. Most importantly, it tells us that areas in the brain associated with these are different. It shows us where to look. It shows us where in the bookcase we can ask, how is LOXP affecting our bookcase? And where can we change it? Thank you.
Okay. All the amazing research that's happening here on campus. Thank you for sharing. Okay, so into our next set of trivia questions. Who held the honor of being the first faculty member on NMU's campus to hold a PhD in computer science? I think it was Dr. Jeff Horn. Thank you for that. Okay, we'll move on to our next question. FDC Willard, AKA Chester, was credited with co-authoring a physics paper in 1975 with owner Jack H. Hetherington. What type of animal was Chester? I think that was supposed to be the start of do 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 do. Oh, darn it. I fell into the trap. Have anybody with a guess? Yes. Where did that come from? Oh, this second row has got all the answers here. Okay, our last question this section. Which animal is known to hug trees to cool down on hot days? Is that a black bear, sloth, koala, or brown tree snakes? Okay, I'll be nice and I'll give you guys this one. It's the koala. Oh. Do we have another question in this section? No? Okay, I think we're on to our next presenter.
Okay, we're going to move on to our next presenter. Before I do so, though, I want to take one more opportunity to thank Tess for doing our trivia today, and Tyler, who worked so hard on getting all of our trivia questions and putting them in order and on the app. And you guys are rock stars. So thank you so much again for your help. Okay, next up, we have Alexia Dalton. And the title of her presentation is Effects of Dextromethorphan on Neurite Outgrowth and Marble Burying Behavior.
Okay, so my study is essentially looking at OCD or obsessive compulsive behavior, and it's really characterized by persistent and repetitive behaviors, and it's often pretty debilitating. So with this disorder, there are various treatment options. However, a lot of the treatment options are about 50% effective, can take months to see any progress, and they have a lot of very bad side effects such as suicidal ideation, sexual dysfunction, and worsening of symptoms and so on. So the idea behind this is to look at novel pharmacological approaches or novel drug approaches to see whether or not we can kind of reduce this behavior or increase neuroprotective effects, which is essentially just protecting our brain cells. So dextromethorphan or DM is known as cough syrup. You can get this over the counter. Dextrorphan is its byproduct. So what dextromethorphan breaks into. And then we have fluvoxamine, which is a common treatment for OCD already, as well as depression and anxiety. So because dextromethorphan is already approved for depression and so on, this would be a more novel approach. So we're looking at this in the cell and marble behavior model. So for starters with the cell model, as you can see, we have our cell body and our neurite, which is just a projection from the cell body, just reaching out, basically communicating with your other brain cells. And so when we add our compounds to this, we should see an increase in the cells as well as an increase in neurites, which just shows from the image that we should already see growth there, but we should see an increase in growth and transmission or communication throughout the cells. Now with the marble burying, we're taking this, as we saw that we a bunch of neurite outgrowth. So then we're taking this and we're putting it in an animal burying assay or marble burying. So essentially, when you take a mouse and you put them in some bedding, about 20 marbles for 30 minutes, they'll just bury and dig excessively. However, when you give them these compounds, then they will reduce the amount of marbles they bury, as well as not reducing the amount that they bury. Oh, excuse me. They will reduce the amount they bury, but they will not reduce the amount of movement they have, which means that the medication is working, but not because that they're just too tired or whatever. So we're comparing this to fluvoxamine because this is already well-known treatment. So then when we give the animals dextromethorphan and dextrorphan, we also find a reduction in the amount of marbles buried. And this is important to kind of further look at different treatments for these types of disorders and kind of carry this on for human trials eventually. And this is the very basis and backbone of this. Thank you.
Thank you. All righty. So many words squished into such a short amount of time. You guys are doing so good. Okay, so our first question for this next little judging break. The sharpshooter, the common name for a group of leaf hopper insects, is known for what trait? Is that camouflage capabilities, flinging their urine, jumping from leaf to leaf, giving John Wick a run for his money in a sharpshooting competition? Now, I guess maybe we can have more than one answer there. Some pretty funny potential answers here. Do we have, did I hear B? B would be correct. Flinging urine. The sharpshooters drink so much each day that they constantly must expel fluid. Scientists are studying the ability of these insects to reduce the surface tension of the fluid bead that they fling with their anal stylus, as it allows for super propulsion. Very interesting phrases in there. Okay, so our next question. Which common farm animal cannot vomit? Are we talking a cow, a pig, chicken, or horse? Ooh, I've heard a lot. I've heard a lot of answers here. I love this. I love to hear some of this reasoning. Okay, let me get a show of hands. Who thinks a cow? Okay, who thinks a pig? Okay, who says chicken? Nice. And who says horse? The horse respondents would be correct. Esophageal muscles only push down, unlike humans, which go both ways. The presence of strong cardiac sphincter prevents anything from leaving the stomach, as food pushes up and out only tightens it. Interesting. Okay, and our last question here on this section is, where were footprints recently discovered that altered the long-held thought that humans first came to the Americas 18,000 to 16,000 years ago? Was that in Alaska? Was it in Argentina, Nova Scotia, or New Mexico? I think I have an idea who gave us this question. Sure, yeah, toss out an answer. New Mexico would be correct. How did that person know this? Who knew this? How did we know? I didn't hear the response, but that was great. So 2 adults and 1 child left footprints in the mud near White Sands, New Mexico. Carbon dating and analysis of quartz grains places these footprints at 21 to 23,000 years ago. All righty, and I think we're up for our next presenter. Are we ready?
All right, our next presenter is R. Ann Flynn, and the title of her presentation is Applying TDA, Finding Secret Holes in Data. And I should probably give her the remote or the microphone.
Hello. So my thesis explores a tool of topological data analysis called persistence homology. So that's a lot. Topological data analysis is using the branch of math called topology, which likes to look at everything as if it is a shape and applies that to data analysis. So we know there's a ton of data out these days, and then more is being created every single day. So the more tools we have to analyze data and the more insight we can draw from that, the better. So while topology likes to look at everything like a shape, it specifically focuses on holes. You can't create new holes, you can't delete holes or get rid of them. You can warp it in whatever way you want otherwise, but holes are the important part. How do you apply that to data? Well, let's say you have a 5-column data set with a bunch of different entries. You can take each column as its own free dimension, and you can plot those entries as if they were 5th dimensional points. So if you've got 100 entries, you plot all those points, you start drawing connections between the closest points, and suddenly you've mapped out a 5th dimensional figure. Okay, so we can apply topology to data by using a type of topology called algebraic topology, which looks at everything like a shape and then immediately reconverts it back into numbers we can actually process via algorithms and data manipulation software like RStudio or whatever else you like to use. So as you can see up here, a KDE diagram is what is spat out once you put it through all of these algorithms in this program. So how does it actually work? Okay, so algebraic topology will look at this shape that we visualized in quotes, because we can't exactly see anything that's got more than 4 dimensions properly, but algebraic topology can, and it likes to walk different paths. And if you can walk a closed loop in a shape, then you can start to make sense of it. And if you've walked a closed loop and there's nothing in there, you've found a hole. And a hole or a gap in data is usually significant for some reason. And that's your next step, is to find out why. So this is a sample KDE diagram from some sample data we've processed. And you can see the line and all the little dots near the line. Those dots are holes that pop up in one dimension and then go away. But the hole up, or the dot up there, is a hole that pops up in one dimension and then persists through many. That's persistence homology. So I'm processing some real world data right now to try to get sense of this very, very new mathematical tool. But we've been processing it for roughly 4 weeks and it's still going, because it is not just 5, it's 50 dimensions that we're processing. There are 200 entries, but this is what we're working on. So thanks for listening.
Thank you so much. Wow. So cool is right. Okay. So what is the name of the beloved robot that would make an appearance from time to time in the math and computer science department? Is it Blaze, R2D2, Kit, or Hal? Do we have any non-math and computer science majors that know this answer? There we go. Blaze. Blaze is a robot made by the late Professor Jeff Horn, used to welcome new and incoming students to the computer science programs. Yes. Good job. All right, our next question. Which country boasts the most biodiversity of any country on the planet? Are we talking Brazil, Canada, Thailand, or Democratic Republic of Congo? Ooh. Oh, Thailand. Good guess. Do we have any other guessers? Brazil. Okay. Is that all I heard? I thought I heard a few Brazils. The Brazils would be right. More than 50,000 species of plants and trees can be found in the lush wilderness and forests. You got it? Okay. No, it's okay. All right, and I think our last question in this section, what country passed a law declaring, so I think that this is pretty interesting in and of itself, but what country passed a law declaring that an apology could not be used as evidence of admission to guilt in court proceedings? I think it's interesting when many of us require our children to apologize for everything. Are we talking England, Mumbai, excuse me, Namibia, Canada, or Norway? I sure hope that that's not the answer. Okay, because I mispronounced that. I don't see anybody that looks like they know. Do we have anybody that knows this? Great guess. Yep, Canada. And I'll have to tell you, the response written here is Canada, Canadians. Enough said. All righty, so are we ready for our next? Okay, thank you guys.
Okay, our next graduate presenter is Mandy Joslin, and the name of their presentation is The Soil Necrobiome, How Human Decomposition Affects the Soil Bacterial Communities.
Researching human decomposition is important for death investigators to better understand how to estimate PMI, or the postmortem interval, time since death. So the time since death can be estimated using things like maggots and bugs or tissue changes, but recent research in the past decade has shown promise with using the soil microbiome as a way to predict PMI. So the soil microbiome associated with a corpse, also known as the soil necrobiome, can be used to estimate PMI, and it's a great source of evidence because there's billions of bacteria in just 1 gram of soil. And also these changes to the soil microbiome are evident over 400 days after decomposition. So what I did for my thesis is collect soil samples from around 5 different donors at the Forensic Research Outdoor Station, or FROST, over time. Then I isolated, purified, sequenced the DNA, analyzed it too, to show me how much is there, or the relative abundance, and who is there, or the biodiversity. So also with abundance, we can see who is thriving during the different stages of decomposition. And using machine learning models with biodiversity, we can also see who is important for predicting the PMI. So the sequencing data, the decomposition observations from the field, and also weather data can tell us the story of decomposition using or through the lens of the soil necrobiome. However, there are some downsides to using the soil necrobiome in death investigation right now. And one of those is that this is a shotgun approach. So a lot of big data over time, a lot of variables, and it's expensive as well. So trying to find out who the important microbes are can make it easier, faster, cheaper for crime labs to use this in actual casework. Also, the second downside is that we just need more research in this field. So more research is needed in different climates and also in different soil types as well. So there's other research going on right now in places like Colorado, Tennessee, Texas. And all of this research can be combined to create a soil microbiome database to help better predict the postmortem interval. So my thesis aims to contribute to the literature in order to have a more robust and more reliable estimation for PMI. Thank you.
Great job. Thank you. What a diverse set of topics. Thank you, guys. Okay, so here we go. First question, Miss Go. What percentage of the Upper Peninsula is forested? %, 84%, 52, or 67%? Anyone in the audience want to guess? Ooh, it's actually B, 84%. Let's see. 8.8 of 10.5 million acres of the Upper Peninsula is currently covered by forests. Our next question, which language is not considered to be among the major Romance languages? Romanian, Spanish, Greek, or Portuguese? Anyone that studies languages here today? A little bit? Okay, all right. So I hear Greek, and Greek would be correct. The official 5 Romance languages are French, Spanish, Italian, Romanian, and Portuguese. What is the most commonly used letter in the English alphabet? The letter A. I love that somebody aligned that with A answer. B would be S. I'm not going to do it like that. E or M? Oh, it looks like we got somebody back there. What do we think? That's okay. E would be correct. Letter E appears in approximately 11% of all words in the common English vocabulary. It is also the second most used key on the keyboard behind the spacebar. Yeah. Pretty interesting stuff. Okay. Are we ready for our next presenter? Okay. Sorry. It's okay.
All right. Will you let me know when we get that next person? You can hear me. We're just waiting on one more submission, so I apologize. I'm supposed to be good at multitasking, but I'm not always so great at it because I'm trying to run scores over here with Lisa and pay attention to when the judges are done as well. So our next presenter is going to be Riley Jensen. And I have to apologize to Riley for spelling your name wrong on the thing that I sent out yesterday. So our graduate student in our office noticed that. So I'm publicly apologizing to you. I felt bad about that. Lisa, do we have all of our submissions for the judges? Perfect. So Riley, the title of Riley's presentation is called animal taxa and microbial soup, gut microbiome diversity in African herbivores.
Good job. We have our slide. Oh, do I just want me to click next on it?
I'm sorry.
Okay.
Here you go.
Inside of you right now are trillions of microorganisms. Each one plays a specific role to help your body function on a daily basis, whether that's releasing hormones, regulating your digestive system, or even controlling your mood. What's even more impressive about these microbial communities is that they function similarly to animal communities all around us. There's predators, there's prey, there's social groups, there's invaders. And just like animal communities, they are impacted by the external environment around them. They're impacted by the external environment around them. Oh my God. Okay. Let's see. Your microbiome changes with your diet, where you live, and even who you interact with. This is also true across all mammal species, including the African herbivores I'm studying for my project. My study area takes place in Etosha National Park in northern Namibia, shown in the map on the bottom right. This park is inhabited by a diverse community of herbivores with a range of body sizes, digestive strategies, feeding habits, and evolutionary histories. You can see these factors and the evolutionary relationships between these species in the middle figure. I'm investigating the impact of these factors on shaping microbial abundance, composition, and diversity. To do this, shown on the right, I extracted DNA from the fecal samples of 11 herbivore species here at NMU's Wildlife Ecology and Conservation Science Lab, then used a bioinformatics program called CHIME2 to classify bacterial groups. And I've discovered some fascinating things throughout this process. Many of these species have host-specific taxa, which are microbes found in no other species. These microbes might exist to help these herbivores survive in this extreme desert environment and might also help them digest plant material high in fiber. Another unique finding is how these microbial communities are shared. For instance, the gut microbiomes of individual elephants become more dissimilar the further apart elephants are on the landscape, shown in the graph and figure on the right or on the left. This is likely because elephants live in very touchy social groups, so elephants in the same herd share more similar microbes to each other than elephants in different herds. So why do we care about the bacteria inside elephants and zebras and wildebeest? Well, because many of these microbes help these animals survive in extreme environments, a loss of bacterial groups could be a disaster as animals attempt to adapt to an ever-changing world. My goal for this project is to provide baseline data about Etosha National Park's herbivore gut microbiomes right now, so that in the future we can revisit to see if any of their microbial communities have changed. We know that climate change and habitat destruction are permanently altering ecosystems around the world. So this microbiome monitoring program has potential for not just Namibia's wildlife, but for threatened and endangered species across the world coping with rapidly changing environments. Maybe the key to overcoming these environmental challenges has actually been inside of us all along. Thank you.
Yikes. Awesome. There we go. Okay. All right, so this next question, how many flowers must a bee visit in order to create a tablespoon of honey? Are we talking 200 flowers, 2,300, 600, or 4,000? I guess I could say 4,000, right? Any bee researchers here? Honey researchers? No? Does anybody know the answer here? Okay, all right. Well, it seems the answer would be D. 4,000 flowers. And they'll fly up to 8 miles in that time, in one direction to accomplish that. So they'll go like a long trip. That's a good question. Okay, our next question. What is the fastest animal alive? And I'll tell you that 4-year-old son of mine would have this answer So fast and so right. Are we talking a tiger shark, a cheetah, a peregrine falcon, or Winnie the Pooh running towards a pot of honey? Well, that sounds like there's lots of us that know that answer. Peregrine falcon, you're right. Let's see, what kind of data do I have here? In a full dive, these falcons can reach speeds of up to 170 miles per hour. Wow, that's fast. Okay, our next question. How often is a new word added to the dictionary? Once a day, once every hour, that's once every other hour, once a week, or once a month? I'll have a little mercy and give the answer to you guys on this one. B, once every other hour. Right? So 4 criterion must be met in order for a word to be added. It's a word that's used by a lot of people. A lot? I don't know. It is used largely in the same way. It is likely to stick around, and it is useful in a large audience. Let's see, I think that's all 3 of the questions. Are we ready for the? Okay, fantastic.
Thank you. We're going to wait for a couple more responses to come in. And while I do that, Tess, is this your phone? We're good? Okay. Well, I still am going to thank our AV. I couldn't have done today without you. So thank you for being here. Thank you for running the Zoom. Thank you for getting all of this working properly. I appreciate it. And then also to Conference and Catering, thank you for providing us with some amazing food and providing the box lunches for our presenters as well. Our next presenter is Riley Lalone, and the name of their presentation is Tribal Relations and NAGPRA, Consciousness, Connectedness, and Cause.
Hi. So when people talk about NAGPRA, they're referring to Native American Grave Protection and Repatriation Act. This is legislation that covers both human rights law and cultural heritage law, and it was enacted in 1990. The purpose of it is to regulate the process of returning Native American remains and cultural heritage back to lineal descendants or tribal communities. This isn't the first protection law. Protection laws peaked in the 20th century and starting in 1906 with the Antiquities Act. However, this is the first law where Native American human remains and cultural heritage was actually protected. Previously, the protection focused on archaeological dig sites, quests for scientific inquiry, and financial investments, really never paying attention to the burials they were digging up or the people they were hurting in the process, both living and dead. The purpose of my thesis is to take into consideration the tribal perspectives of NAGPRA law, whether the law that was written by the federal government actually helps or hinders the processes of reunification for tribes. There are 3 aspects I look at: consciousness, connectedness, and cause. These are drawn from Sean Wilson's research is ceremony, where he looks at what's called relational accountability. This is the act of maintaining the right ethics between research participants and the broader communities. So this is one of the integral parts in Indigenous studies. The purpose of Indigenous studies also includes taking into consideration and recognizing the knowledge of colonization, recovering the Native American, understanding the idea of manifest destiny, and the struggle for self-determination. My thesis does all of this taking into consideration the tribal voices of the voices of tribal nations across the United States. Thank you.
Thank you. Say again, it's really important work that's happening here. All right, so first question for this little break. Where is the world's largest national park? Is that Banff National Park in Alberta, Canada, Cradle Mountain in St. Clair, Tasmania? Are we talking Northeast Greenland National Park in Greenland or Torres del Paine in Chile? Do we have anybody that's been to all of these? Ooh, do we have anybody that's been to any of these? Nice. Where did you visit? Banff. Okay. Where did we visit? Banff. Okay. Pretty cool. I think I'll just tell you because it looks like we've got some pretty confused faces here. C, Northeast Greenland National Park is the largest national park and the 9th largest protected area on Earth, spanning 375,000 square miles. Pretty big place. All right. And what is the term for the physical reaction of goosebumps when experiencing a strong emotional response from art, typically music? Are we talking the Babinski sign, frisson, occitaency, or pharyngeal reflex? What was that? A, the Babinski sign would not be correct. I'm sorry. The answer would be B, frisson. Studies show anywhere from 55 to 86% of people are able to experience this sensation. The wide range is likely due to the tendency of some people to feel frisson in a higher rate than others. Interesting. All right, I think we've got one more question in this section. Which professor earned a patent in 2007 for their resource sharing algorithm? I think it was Dr. Jeff Horn. Righty, I think we're on to our next presenters. What? A brain break? Well, let's do it right now. Yeah. What's the brain break? Oh, I was just thinking we might need a little bit of brain break. We've been sitting for a little bit. If anybody would like to join, I'm going to say let's do a deep breath and stretch up to the air. And just feel free to stand up and move if you so choose. Your brain needs it. We all know it. So you can pull in more great information. Let's shake out. If you need any water, grab a drink real quick. Yeah. Play with your fingers. Yeah. Next year we'll take more of a break.
Next year, I promise to put one more break in there for you guys. We had a little bit of a break in between the McNair graduates And we've had several graduate presentations with no breaks. So next year, I promise to put one more in there. But I think that we are ready for our next presenter. So our next presenter is Alexander Adamer. And the name of his presentation is Epigenetic Factors and Post-Traumatic Stress Disorder.
Alrighty, so previous work in our lab has shown that the novel combination of the SSRI fluoxetine and the dissociative anesthetic ketamine were shown to decrease fear responses in a conditioned fear paradigm within mice. So my work within our lab will be looking at how do these pharmacological compounds interact with the epigenetic factors of GABAergic metabolism within the central fear memory circuit, and this is within the context of PTSD. So within PTSD, there's a significant decrease in an individual's ability to dampen fear response and dampen fear memory. And the fear memory circuit itself is made up of a dynamic interplay between excitatory and inhibitory neuronal populations in the medial prefrontal cortex, the amygdala, and the hippocampus. So GABA is the major inhibitory neurotransmitter in the central nervous system, and it's synthesized from the major excitatory neurotransmitter in the central nervous system. And this is carried out by GAD67. We know that in PTSD, GAD67 and GABA are significantly underexpressed, which lends to symptoms such as emotional reactivity problems and things of that nature. So we also know that fluoxetine and ketamine are able to increase GABA and GAD67 in this brain region and kind of balance out that inhibitory, excitatory balance, if you will. But we don't know exactly how this is happening, although we do know, and it's been suggested by other research, that epigenetic factor known as HDAC1 is significantly overexpressed in the hippocampus and binds directly upstream from the GAD67 gene, likely blocking GABA and the lack of inhibition. So our research will use Western blot to determine if these drugs are inhibiting the epigenetic factor and to see how much GAD67 is present, because we would expect that if the epigenetic factor is inhibited. We'll also use confocal microscopy to determine the subcellular localization of this epigenetic factor. And all of this is in hopes that we can inform future clinical trials with these drugs in the context of PTSD. Thank you for your time.
Thank you. It's so great to see the relief when they hand this microphone over, each and every one of them. It's like, oh, please, yes, thank you. You guys are doing a great job, really. Okay, so first question in this section, who designed the Vietnam Veteran Memorial in Washington, D.C.? Oh, somebody knows for sure. I like that. Does this person, I didn't hear who said that. Can you share more about Maya Lin, if you're familiar? Okay, I'll tell you the little bit that I see here. The winner of the Medal of Freedom was an undergraduate architect student at Yale University when she won against 1,400 competitors with her memorial design. All right. Okay. All right. What unassuming farm animal can be used to guard against coyote attacks? A goat, a llama, a donkey, or the rabbit from Monty Python? Yeah. Ooh, we got lots of opinions here. Okay, 3, 2, 1, let's hear it. A donkey would not be correct. Okay, what's the next one? Llama. Studies prove that even one guard llama can be effective. I like that. Still have my eye on that rabbit though. Okay, and then I think our last question for this section, what fruit was used as a status symbol by the English in the 18th century? Was it oranges, lemons, mangoes, or pineapples? Oh, good. Yes, pineapples. The high cost of transporting the fruit to England drove the price higher than most could afford. Wealthy individuals would carry their pineapples with them or display them without eating them. Oh, yeah, just maybe that's where the whole hat thing came from. Yeah, very interesting. I think that's all that I have for my trivia questions right now. Are we ready?
We're in. Okay, I should probably grab my clicker, which I was so excited to call AV this morning and say, hey, do you have one of those clickers that would make my life easier? And then I keep forgetting to click over. So that's just the story of me. Our next presenter is Yusuf Kwadumi, and the name of his presentation is The Effects of Transcranial Direct Current Stimulation on Hand Grip Endurance in Adults 60 years or older.
All right, good. All right, so we are, I'm sorry, currently there are 49 million Americans who are age 60 years or older. And as we age, we experience neuromuscular changes that have negative impacts in our motor function as well as functional independence. Transcranial direct current stimulation has been linked to improvements in motor function. So for this study, we stimulated the prefrontal cortex, which plays an important role in exercise regulation. So the prefrontal cortex processes internal and external cues such as, rating of perceived exertion or knowledge of exercise endpoint. So your brain is like a muscle, all right? And just like any muscle, it can fatigue. So with this study, we hypothesized that by stimulating the prefrontal cortex, we would delay that fatigue and therefore delay exercise termination, allowing individuals to exercise for a longer period of time, and therefore they can reap more benefits of exercise. So what we did was we had individuals who are 60 years or older with no underlying conditions come in for 2 sessions. In a randomized order, they either experienced the stimulation of the prefrontal cortex for 20 minutes, or they experienced a fake stimulation. So we turned the stimulation off after 30 seconds. So in this image here, the cap here kind of shows us where to place those electrodes to stimulate the prefrontal cortex properly, as well as holds the electrodes in place. And then they squeezed a hand grip dynamometer to measure their maximum force. Then after stimulation, they did a hand grip endurance task where they squeezed. We saw how long they could squeeze and maintained more than 50% of their maximum. So data collection is still ongoing, but preliminary results show a significant difference between the stimulation and the sham or fake stimulation group. And the take-home message here would be that, we may be able to provide older adults an opportunity to exercise longer and reap more benefits, preferably in a rehabilitation setting. So if they were to have a knee or hip replacement, we could stimulate them before the rehab session and accelerate their rehabilitation process, getting them to, functional independence quicker. Thank you for your time.
Thank you. right, so we've got some more letters mixing up. I'll be a little more smooth this time, okay? What are the only two letters not present on the periodic table of elements? Is that J, R, V, Q? This is multiple correct answers. Yes, we are trying to trick you, not really. Yay, J and Q. Very quick to get that. Good job. All right. Which fact about Lake Superior is untrue? Lake Superior was formed roughly 1.2 billion years ago. It takes 551 billion gallons of water to raise the water level 1 inch. The shoreline of Lake Superior is 2,726 miles long. There are 20 different species of fish within the lake and its corresponding waterways. Which of these is untrue? I heard D. D would be correct. There are 88 different species of fish held within the lake and its waterways. Okay. What US president once sued a Michigan newspaper for libel or defamation? Was that Abraham Lincoln, Grover Cleveland, John Tyler, or Theodore Roosevelt? D is, ooh, interesting. Yes. Huh? D, yeah. D is correct. I'm sorry. My response, my answers are a little different here. Had to make sure. Yes. Theodore Roosevelt. He sued a newspaper called the Iron Ore for 6 cents, which he is quoted saying was the cost of good newspaper. The paper accused him of public drunkenness, and he won the case. Very interesting stuff. All righty. I think we've got them all in. What was that? Oh, it's all good. There will, if any of you did participate online, am I correct? We've got I think we have a prize. I think we've got a prize for the winner. I hope we do, because I just said we did.
Thank you again, Tess, for helping us with trivia today. We have one more presenter today, and that's going to be Megan Wells. And the name of Megan's presentation is Drug Treatments for Post-Traumatic Stress Disorder in a Mouse Module.
Hi, everyone. So today I'm going to be talking to you guys about drug treatments for post-traumatic stress disorder within a mouse model. So to start, just some background information on PTSD. We know that this disorder forms after exposure to a traumatic event. So that's going to be something like a car accident, an assault, so on and so forth. We also know that symptoms of this disorder are severe and are extremely detrimental to someone's quality of life. Current pharmacological treatments for PTSD have been found to have issues in response and remission rates. So they're seeing that fewer than 50% of patients are fully recovering from the disorder following treatment. That emphasizes our need for new treatment options. My thesis is going to be looking at 2 different drugs. So we have fluoxetine, an SSRI, typically used for something like depression, as well as ketamine, a dissociative anesthetic. I'm going to be looking at the effects of these drugs when used alone and when used in combination in regards to PTSD, as well as generalized anxiety disorder. So to do this, I'm following an established fear conditioning model in which I use an aversive stimulus to induce a traumatic experience in mice. 4 hours after this experience, mice will begin treatment. So they're either going to get saline, or ketamine, fluoxetine, or a combination of the 2. We also have a group of mice who will not undergo any aversive stimulus and will not receive any form of treatment. So they're going to act as our control group. 24 hours after that aversive stimulus, as well as 2 weeks later, I'm then assessing the mice for certain behaviors that are known to be associated with PTSD. Also occurring 2 weeks after the aversive stimulus is a test that we run the mice through to look for anxiety-related behaviors. So with my results, I'm going to start with this graph down here at the bottom. So that is relating to an anxiety behavior. And with that, we found that the fluoxetine only treatment group was actually on par with that control group that did not undergo the aversive stimulus. So that's going to indicate to us that that fluoxetine only treatment was most effective in reducing anxiety. When we look at these two graphs here on the side, those are going to be associated with two behaviors that relate to PTSD. And we actually had very similar results for these two behaviors. So looking at 24 hours after the aversive stimulus, which is going to be this and that, we see that all treatment groups are engaging in high levels of that behavior and therefore experiencing high levels of fear. But when we looked at it again 2 weeks later, we saw that that combination ketamine and fluoxetine group had significantly fewer behaviors. So that's indicating to us that that combination treatment was most effective in reducing fear, which as we know is a huge part of PTSD. The last point I want to make is that all of us are at risk for developing PTSD or being affected by it. Whether it's ourselves or a loved one, it just takes one situation. That's why it's important that we're looking for a safe and effective treatment option. Thank you.
Thank you very much. Okay, we are going to open up the People's Choice voting now for graduate students. So I'm going to leave this QR code up on the board. The other thing we're going to do right now is we're going to add up all the judges' scores. So we're going to wait for those to come in. And we're going to take kind of like a 5-minute break. And in 5 minutes, we will come back here and we will have our Q&A session. So we would ask that all of the presenters come up here and we'll just kind of hand a microphone around. Are there any questions before we take a little bit of a break? All right. I would also like to remind everybody that we have a bunch of food over there. Please go help yourself if you have not. And to all of the presenters, looks like we have quite a few boxes of presenter lunches left. So make sure you grab yours on the way out, please. And the food is good.
So try it.
We can bring. Thank you, Lisa. I just feel like it's gonna take like almost all of our audience away if we have a Q&A.
Okay, I agree, because I'm not looking behind me, so I
Okay.
don't see what's back there. If that's what you, that seems like the right thing, like that's what you should do. Don't listen to me. I'm just sitting here adding. For some reason, this one.
What do you think? The best way to do it, have them come up here or? Okay.
Yeah.
All right, and then we have one more microphone to pass around to people who have questions. Okay, at this time, I'm going to ask all of the presenters to come up here and stand again, just in case we have any questions. We're still working on getting the scores tallied so that we can announce our winner today. Winners. And we're still waiting on some people's choice voting to come in. And I knew that when you guys all came up here that it would probably take most of our audience away. So first of all, let's give them a big round of applause again, because I think they did an amazing job. They had to memorize everything that they talked about today. They couldn't use notes like I had cheater notes up here and still messed up. They couldn't use notes at all. So they had to memorize everything. And it just shows you how hard they've been working. So thank you once again for participating. At this time, are there any questions for any of our presenters? All right, hold on. I'm looking for my other microphone. Here it is. I'm gonna leave this with you, and just in case somebody needs it. I'm gonna come to you first and then you.
I don't know if Thanks. I've got a question for you. With the people over 60, have you looked at applying this? My dad just had a stroke, and it was in the front of his brain. Have you looked at applying this to the recovery of stroke victims as they recover their physical abilities?
Yeah, so there's been a bunch of research with like stroke victims and like Parkinson's. I'm actually applying to PhD programs right now where I'm looking to work with people suffering from Alzheimer's. So a lot of like clinical populations are being looked at. My focus was mainly, and that was kind of my gap in the research, is nobody's really looking at exercise performance in adults over 60. People were looking at, clinical populations as well as like younger healthy adults in exercise performance. But what about like the healthy adults who, maybe are, hey, you have like, onset atherosclerosis, let's start exercising and, trying to gain as much as they can before, we start to see, drastic declines. So that was kind of like my focus in the research.
I have a question for you also.
Oh, sorry.
Are there any, have you found any by exhibiting, is that negatively impacting or
and that's kind of why I chose like hand grip, because it's very, non-fatiguing. You can have them, squeeze like as hard as they can, and then, they walk out like non-fatigued, right? So future research could like at like larger muscle groups and kind of start to incorporate other variables on, are there, any detrimental effects to this? But this type of stimulation I chose was anodal stimulation. So that's like the electrode is going like specifically over, you have an anode electrode, which is linked to like the brain plasticity, like improvements in motor function. And you have like the cathodal, which is, has that opposite effect. And so the stimulation that I chose was anodal over the prefrontal cortex, more specifically the dorsolateral prefrontal cortex. And people do respond like differently. So that is a limitation of my study is, if I could go back and do it again, I would have like a anodal group, a cathodal group, and a control group. But when you present like, hey, do what 60-year-olds want to come in and voluntarily, stimulate their brain, getting them back for 2 sessions has been a struggle. Getting them in for 1 session has been a struggle, and then definitely for 2. So if I just had like more access to like an older population, that would be like how I would go about that, if that answers your question.
You had a question?
For Flynn.
Okay.
With the 4 dimensions, like the data sets, and then the 5th dimension with the dots, do you have any examples of like the data sets and the dots to kind of like a generalized explanation to kind of give some context?
So the diagram I presented was from just a sample data set with 5 different variables or that 5th dimension. It's a little bit difficult, as you can imagine, to cram a bunch of different examples up on one slide. But we are processing some sample equations that we know the look of and we know the expected outcome of that are in the limited dimensions that we can see. And then the ones that I'm processing that I hope to get some insight on are, I'm doing a 30 column and a 50 column. And so a 30 dimensional and a 50 dimensional shape that we're processing. I wish I could present those shapes in any modicum of understanding that we can just look at it and see. But we can really only see up to 4 dimensions in a graph.
I guess what would the outcome set be? Like what would the group look like of objects?
Okay. So the outcome wouldn't change the data at all. This is a way of analyzing any pockets that we can't necessarily see right off the bat in data. Like persistence specifically has not really been implemented much yet. It's extremely new, at least as far as math goes. It's less than 15 years old, and that's very, very new for math. But TDA, where it comes from, has been used to help find cancerous tumors before doctors are able to see them just by looking at x-rays, for example. And so while it wouldn't change the data itself, those gaps and like the one significant dot I showed, those gaps that you can find could lead you toward analyzing the data and finding insights from that. But as far as actually changing the data itself, it doesn't do that.
Okay,
I hope that answered your question.
Yeah.
Thank you.
Thank you.
Do we have any other questions? Oh, right up here.
I hope to study conservation biology and animal behavior.
I hope to go on to a PhD program, either studying medicinal chemistry or pharmacognosy, one of the 2. Thank you. Thank you.
Every one of you. Amazing. Thank you very much.
Thank you.
Do we have any other questions? Oh, Dr. Eckert.
Challenges in working with whether they were animal or human. Curious about the lab animals, what some of the challenges up there after working with subjects, what that experience was like.
I think for me, it's always an ethical question because I'm working with mice. And so we ran a power analysis in my lab to figure out the minimum number of specimens that we would need to properly answer the questions that we're asking. And unfortunately, that still ends with 60 specimens that are needed. And I know in other labs, actually, that's a pretty small number compared to other tests like in pharmacology labs. But As a way to deal with that, I actually started working in the zoology museum, doing dissections, and started volunteering doing dissections before I even started working with my specimens, so that I could hopefully reduce the number of specimens and reduce harm, essentially. So it really was about mitigating harm as much as possible. And that just took a lot of personal time practicing dissections before I ever worked with an animal that we are raising for this project.
For my project, I interviewed tribal historic preservation officers, and the history of research with tribal individuals has not always been very happy in the past. So I did face a little bit of apprehension from them, understandably so. Being a tribal member, it was a little bit easier, but there were a lot of questions about what does research entail and how is the research that I'm doing changing from the research that has been done before.
Yes, like I said earlier, it was very hard to convince 6-year-olds, sorry, to come in and get their brain stimulated. But I guess like a positive note is like they always have the best stories to tell and like the best life advice. It's while they're getting their brain stimulated for 20 minutes, it's a nice little like just glimpse into their life, and they love coming in and just being around me and my research assistants and talking with us. And I guess the hardest part is knowing like after they leave is I might not see them again, and like in the 2-hour session, like the kind of I guess relationship so to speak that we've built is it's just a good time having them in there. So like that'd probably be like the hardest part is getting them in and then like kind of getting them to leave.
So I actually work with microbes mainly, and the hardest part I'm facing right now is culturing one of the fungal, the botrytis cinerea, so the fungal pathogen, because my professor had some of it in stock, but then it's like an old stock, so we're trying to regrow it, and it's just not going. And it's not like a human who's gonna be like, hey, like you need to give me some more nutrients, man. It's like you got to keep trying different methods to see what they like best. So that's my difficulty with it, but it's quite interesting. It's a similar process. Personally, I'd say I need the presentation experience. So thank you for everyone who came so I could talk in front of everyone, because I plan on doing other research conferences in the future. And so this is a great experience for me because this was my first time presenting my research in any kind of manner.
Great job.
I definitely agree. I've presented at the McNair scholarship, and that was a 10-minute talk, which is a lot different from a 3-minute talk. So it was very interesting to take a summer's worth of work and put it into 3 minutes, and it was a lot of fun. Thank you.
I found it really interesting to try and condense my research into 3 minutes because there's so many factors that go into it and steps that, I mean, it's a 3-minute speech and it took me like 2.5 hours to write because I'm like, how do I make this make sense and also include all of the important things? So I felt like it was kind of nice, I guess, to be able to learn how to condense it and get it across to people who aren't in my field.
Yeah, I'd go along with that point. It was really good to try to condense things and use as little jargon as possible and somehow get better at science communication, which can be surprisingly hard. And it was really fun to present as well.
As a math person, it can be a little isolating sometimes to do research because you don't get to go out there and work with living things or things that are directly integrated into the world. And so it's very interesting, but it can be a little bit detaching. And so to come here and see what people are doing with their own research that uses tools that math had, I'm sure, a hand in developing at some point. I mean, not me directly, but it is connected. It's just, it's nice to see that it will happen it's nice to see that it will play a role somewhere, and to see all of the things that you guys are doing is really, really cool. And so that was my favorite part, was get to see all the variety of what's being done right now here. So it's made me really proud to go to Northern. Woo, go Cats.
Go Cats.
I would say probably definitely the practice with public speaking. And then in addition to that, trying to cram like months and months of research into 3 minutes is very challenging, but it went well, yeah.
I think it's really valuable for me to practice explaining my research in a way that like everyone can understand, especially for my future career as well in forensic science. So that way, you know, I can communicate with someone who may not be a microbiologist and be able to communicate my findings. So.
Okay, I'll go too. I think, you know, just practicing is always good, obviously. And then being able to like hear what everybody else is doing is cool, but also just being able to share. I feel like the Native American studies is kind of like this blip of studies out in the middle of the universe that not a lot of people know about. And there's a lot of intersectionality where it crosses with a lot of really other great fields that you can study. And so, yeah, that's cool.
Yeah. I think this has been a good exercise in memorization techniques, because I clearly do not give public speaking like talks like this ever. So it's just interesting to see like work on those techniques. I was supposed to last summer, but the scheduling didn't work out with our collaborators down there. So unfortunately, it kind of got like canceled last minute. So, but in the future, I really hope to make it to my study areas.
Can you tell us a little bit about how you got your samples for your research?
That was a process. So we are working with a team, or we worked with a team in Namibia who collected these fecal samples non-invasively. So basically, Namibia is a really vast desert landscape. And so what they did was they would wait in blinds for hours at a time and just wait at these water holes, because all of these animals come to drink water at these water holes. And they would just wait there for hours for a specific animal to poop. And then after the animal left, they would go out and collect that fecal sample. And then one of the most challenging processes of my research was actually just getting those samples to the US. So 3 of my species are on the endangered species list. So we need to go through CITES, which is the Convention on the International Trade of Endangered Species. And so we were just waiting on permits forever. And then there was some logistical nightmares with getting it here. So working on in a separate country, like working in collaboration with a different country and collaborators from there has also been kind of an eye-opening experience.
All right. Do we have any more questions, Tess? Oh, I'm sorry.
Okay. I mean, I really liked being afraid, honestly. I think I've the more I study the brain, the more I've developed a different relationship with the brain, I think, wherein it's just an organ. It's separate from myself, and it's easier to challenge myself. And I lean into those negative feelings a lot more, and they just become an experience. They're just passing, they're just there. And this is such a fun experience wherein it's wonderful to be afraid. Of course, I'm afraid to eat a lemon bar up here when everybody else is just standing here. Of course, I'm afraid to be the first presenter, but it's a great type of fear. So honestly, thank you guys for letting me be afraid here. It's a wonderful exercise. He didn't get to share his either, I don't think.
Oh, my favorite part was when I handed the mic back. But no, this has been a great experience. But my real favorite part is just seeing like what's being done on campus, like the awesome research that's being done by not my peers and colleagues, I guess, if that's the right term to use there. But yeah, some really cool stuff that's going on campus, and just being able to just try and soak as much in as I can. It was a great experience. So. Yeah. Mhm. Yeah. It's always a great, oh, sorry. It's always a great time to see your graduate studies staff. So especially my man's Tyler. Shout out to Tyler.
All right, I think that's going to conclude our question and answer section. We are going to be announcing the winners very, very, very shortly within just the next few minutes. So if you guys want to have a seat, again, thank you so much. It's a great day to be a Wildcat. Do we have the winners?
Okay. All right, we got the winners. Don't read the scores.
So this is third.
Oh, I do. I have that.
Okay. What? I think we have a kid. Okay, before I make the announcement of who won, I do have those winners in my hand. I'm going to go ahead and say just a few more things. I'm sorry, I need to get into my computer. So this is just a few of the programs that we offer in our College of Graduate Studies and Research. Be sure to check us out online. We have lots of online options, and we also have lots of in-person on-campus options as well. And then you can also combine the two a little bit. So we also have a great program where if you don't see something on the list that you like, maybe you can pick and choose and build your own program as long as it follows certain guidelines that we have set forward. Okay. Before I announce the winners, there's just a few more things I want to say. Thank you, everyone who participated today. Thanks again to our wonderful judges for taking time out of your busy schedules to be here and participate in this. I'm super proud to have you here. I'm super proud of all of our students who participated today. I know it takes a lot to get up here and stand in front of everybody. I mean, I'm hiding behind a podium and they were out there in front of all of you. I'm back here in my little safe zone. We do have some events to look forward to. I just kind of wanted to point those out before we announce the winners. We have a student celebration of student scholarship coming up on April 18th. That's an amazing opportunity for all of the presenters in here to take the same research and apply it to that celebration. It's a big day. It takes place over in Jacob Eddy, and it's an all-day much Jamrich, that's what I meant, not Jacob Eddy. Jay, that's where my mind was. Takes place over in the new Jamrich, or new to me. And then we also have the buzz session coming up on March 14th. And so be sure to check that out as well. So I believe this is the moment that you've all been waiting for. I'm going to have Lindsay come up here and announce the McNair winners.
So these are great odds. Love that there's 3 opportunities for fabulous prizes of money and 2 students. So our number 1 person, first place, Caitlin Younglove. And then our second place is Sabrina Matta, and our people's choice of the 2 is Caitlin Younglove. Woo to both.
we are going to be mailing you your certificates and also your prizes, okay? So we just don't have those here today because we wanted to be able to get your names on those. So we will be sending those out to you. We'll be in touch. Finally, we have our grad students. Third place is going to go to Yusuf Kadumi. Second place is going to go to Alexia Dalton. First place is going to go to Megan Wells. And then we do have a repeat. So People's Choice is also Megan Wells. So congratulations again. And again, to the grad students, I will be reaching out to you and we will be sending your gifts. Megan, I will be in touch because you will be going to the MAGS conference. So congratulations. All right. Thank you for joining us here today. And that's going to be it for 3 Minute Thesis, 7th annual in 2024. Thank you.